A very good afternoon to all of you, ladies and gentlemen. It's my pleasure to welcome you to the Nanostructures Enabled Direct Communication to Living Cells, the online talk which is jointly organized by the International Relations Office together with the Chinese Academy of Sciences. I'm Roshan Dept from the International Relations Office, and I'm happy to be your host in today's program. The objective of today's program is to introduce about the cutting-edge research in the field of micro-nanotechnology for biomedical applications. So before we start, a small announcement for all our participants. So please be kind to post your questions in the chat section below, and we are very happy to take them after the presentation. And please be kind to switch off your audio and your video during the presentation. So with that, now I first invite Dr. Damayanthi Herath from the Department of Computer Engineering of the Faculty of Engineering of University of Peradeniya to introduce the resource person for today's program. Over to you, Dr. Damayanthi.
Thank you, Roshan. Good afternoon, everyone. So happy to be in this audience today, and I'm looking forward to the talk. Let me warmly welcome and introduce the resource person for today, Dr. Yuhong Cao. Dr. Yuhong, who will be speaking to us today, holds a PhD in Material Science and Engineering from the Stanford University. She holds a Bachelor of Science in Chemistry from Linfield College, USA. And she has got her PhD in 2018 from the Stanford University. And it's been on development of a non-destructive nanostraw system for longitudinal living cell sampling with Professor Nicholas Melosh. She has had 2 postdoctoral trainings, working with 2 Nobel laureates, one at University of California, Berkeley, and the other at the Stanford University. At University of California, Berkeley, she has worked on T-cell engineering by applying CRISPR-Cas technology with Professor Peidong Yang, who is a fellow of National Academy of Sciences in USA. And there she has partnered with Professor Jennifer, who is a Nobel laureate in chemistry. Her postdoctoral training at the Stanford Uni has been with Professor Stephen Shu, a Nobel laureate in physics. Currently, Dr. Yuhong serves as a professor at the National Center of Nanoscience and Technology, Chinese Academy of Sciences, and she has been there since December 2020. With regards to her research, Dr. Yuhong's research focuses on overcoming the current drug delivery barriers through micro-nanotechnology. And such work is anticipated to produce big impact in various biomedical fields, including immunotherapy, regenerative medicine, and animal disease modeling, leading to groundbreaking scientific discoveries. And of course, to learn more, let me warmly welcome Dr. Yuhong for the talk. Over to you.
All right, thank you so much. Yeah, so should I share my screen now?
Yes, please. Yes.
All right, let me share my screen. And okay. Oh wait, 1 moment. I need to turn off my video because my signal is not very strong, and I'm worried that during my talk, I got kicked off. Yeah, so it is my great pleasure to be here today to present my work, and thanks everybody for inviting me to speak. I'm an engineer by training, a materials engineer, and I have a lot of interest in biology and nanotechnology. And as a material engineer, I deeply believe in the advancing technology will have a great impact in life science. So today I'm going to talk about how do I apply the nanomaterials to advance the biological research. Let me first give you a overall picture of my research interests. My overall research interest is in intracellular delivery. It is the essential step in biological research and therapeutic applications. So for those of you who are not familiar with intracellular delivery, it is the methodology to safely send molecules or materials into the target cytoplasm or cells, so that we can engineer the cells or study the cellular behaviors. It is involved heavily in our biological research, and in cell therapy, we also use intracellular delivery for engineering our own cells or the donor cells to fight diseases. And in bioproduct manufacturing, delivering messages to biological machinery such as bacteria, yeast, and plants would allow us to manipulate the cell function so that we can produce the desirable biological compounds. And this is my overall perspective. So intracellular, to me, intracellular delivery is a very big concept. So because the physical morphology and chemical barriers are different between various types of cells, there's no one size to fit all strategy. So we always have to build new systems to meet the current experimental needs. So my research is particularly interested in delivering or engineering novel delivery strategies for particularly in vivo cell engineering, in vitro oral in vitro oral delivery, and zygote engineering, and tissue plant, especially plant tissue engineering. So in this talk, because we have a short in time, I'm just gonna focus on in vitro intracellular delivery. So currently we have established many delivery systems, and here I list the most popular one that we use in daily basis. The viral vector basis delivery, chemical based, and whole cell electroporation. Although they are very powerful and useful, they suffer from various kind of limitations, and which actually slows down or even blocks the progress about biological discovery. And here I list the most the challenges of each delivery systems. For viral vector, it suffers from high risk of insertional mutation, and for chemical-based delivery, it is inefficient to primary cells. And for whole cell electroporation, it has high toxicity. If you really use that before, you would notice that the cell after the electroporation, more than 50% cell can be dead, and it was and the cell dead body are all floating on the liquid surface. So besides these challenges, there are other challenges in the system. For example, length of dosage control, and non-universal delivery, and single shot delivery. And because of this, this imperfection of the current delivery methods motivates engineers like me to develop the next generation intracellular delivery systems to facilitate biological research study. So over the past few years, I developed two novel systems, which are the Nanostraw system and the nanopore electroporation. I will talk about these two systems in more details during the rest of my talk. So first of all, why do we want to use nano? I think the first question is, why do we want to use nano? So one of the biggest advantage of using nanomaterial is its size is small. So that is this is compatible to a cell. So if you compare their size with a cell, you will notice that they're almost invisible to a cell. So the extremely small size of nano devices make it relatively easy to go in and out of the cell without causing too much damages. And the other advantage in nanostructure is that it can enhance the local physical phenomenon. For example, mechanical strength or electric field strength can be greatly enhanced to promote cell membrane penetration. At the local, and I will discuss this in more details during the talk. I'll put examples. In the next 20 minutes, I will talk about the two particular systems. One is Nanostraw system. First, I will talk about Nanostraw system, and then I will move on to talk about the nanopores. I will first talk about how do we design it, and then how this design will give unique feature of the Nanostraw system. And then I will talk about the concept of living cell biopsy, and how the Nanostraw allows longitudinal living cell sampling. So when I started with my graduate study and decides to build the next generation delivery system, I thought very hard about what kind of system that I want to build. So instead of just build a non-viral system like we currently have that would directly dump a random number of cargo molecules in cells, we hope that the cell we can really send correct messages, and we can build a channel that allows us to directly communicate with cells with high accuracy, and we can repeatedly send in precision messages to cells and also receive their feedback information. So the idea comes, the very initial idea to build the system is through the inspiration by the protein channel on the cell membrane. This nanoscale intracellular junction provides fluidics access and promote molecular exchange. They're small, and they're also small enough to avoid cell toxicity. We were thinking about if it's possible to build an artificial channel like a hollow nano tunnel or a nano needle to mimic the protein channels. So that we can directly manipulate and monitor the gene expression in living cells. We started with, to fabricate this structure, we started with the membrane with pores on them, and then we use atomic layer deposition to deposit aluminum oxide coated on the entire surface. And then we use the reactive ion etching to etch off the top layer of alumina, and then use oxygen plasma to selectively etch off the substrate polymer to reveal the nanostraw. And here are the scan electron microscopy image of the nanostraws. You notice that the opening of the nanostraw is about 100 nanometer in diameter, and the height of the nanostraw is about 1.5 micrometer. And there are two other images showing that the cell is cultured on the surface of the nanostraw and interfacing with the nanostraw. So if you do the counting, you will notice that for a cell like a HeLa cell, there are about 20 nanostraws that is interfacing with a cell. So we have made this structure, and the next question is, what is the condition for the nanostraws to pierce the cell membrane? So we did it's not simple, but we did a simulation and calculation to predict how much force we need for the nanostraw to pierce the cell membrane. And this is a very complicated calculation, so I'll drive into the conclusion. So what we find is that for a typical nanostraw in diameter of 150 nanometer to pierce the cell membrane, it requires a force of 0.8 to 1.5 nanonewton. And we also find that the gravity, if we use gravity, the gravity of a cell will give the cell for about 10 to 100 piconewton. And which is actually smaller than the 0.8 to 1.5 nanonewton. And we also calculate the cell membrane and substrate binding forces, which is the cell membrane that attached to the surface, the force coming from the stretch force. So what we find is that the stretch force coming from the cell membrane and substrate binding is about 0.1 to 5 nanonewton, which is approximately larger than or equal to the cell membrane failure threshold. So the conclusion that we got here through the calculation is that in order for the cell membrane to be pierced by the nanostraw, the cell membrane needs to be tightly attached to the substrate. So knowing that through our theoretical calculation, knowing that the cell has to attach on the surface, and then we did a small test to prove the concept. So in this experiment, after we allow the cell to adhere on the surface and making good contact to the nanostraw, we delivered the cell membrane impermeable azide sugar into the cell through the nanostraw. The mechanism is simple. Upon successfully entering into the cell, the azide sugar is enzymically converted to the sialic acid group and incorporate onto the cell membrane glycoprotein. And and the group retains the azido motif, which can specifically labeled by the DBCO fluorescence through the bioorthogonal reaction. So if there is enough delivery, we should be able to see fluorescence accumulation on the surface of the membrane. And indeed, our result shows that with Nanostraw, well, after 48 hours of incubation with the sugar, the cell that culture on the Nanostraw shows signal accumulation on the cell fluorescence for signal accumulation on the cell membrane, whereas the cell that culture on the flat surface didn't seem to have so much signal accumulation. We also check the intensity by doing this intensity analysis. This is so if you look at the line trace showing in this image, you notice also that the fluorescence signal accumulation from the cell on the Nanostraw is much higher than the negative control. But notice that so the conclusion right here is that Nanostraw really allows direct intracellular access. However, if you look at this condition, we use 48 hours, which is too long and it's certainly not efficient for normal bio research experiment. Then we will move on to just to modify or improve the system to make it more to increase the mass transporting efficiency. So in order to do that, we need to go back to analyze the mechanism why we have such low delivery efficiency. So if we check the mechanism, delivery mechanism, you'll notice that it is the delivery mechanism right here is diffusion. If you learn some chemistry or physics, you know that it is through the random walk mechanism, and it is just like bouncing back and forth. The molecule has to bounce back and forth many times until you reach to their target. So if we want to improve the transporting efficiency, the simplest way is to apply some force to drive the cargo into the cell. What we did is just to add a really weak voltage potential, about 10 volts, so across the straw to electrically drive the charged molecule into the cell. We also did a simple calculation for typical highly negatively charged molecule like mRNA. The electrical driving mass transporting is about 100 times faster, or more than 100 times faster than it is through the random walk mechanism. Equipped by the electric field force, we are able to transfect various kind of molecules and drive the relatively large mRNA into the cell, and we got very good transfection efficiency. We did in this experiment, we did tested a few primary cells like this one, the iPSC-derived cardiac cells, and human stem cells, fibroblasts, mouse glia cells, and mouse primary neuron cells. And this bar graph shows the actual efficiency. The efficiency is about 85% for human fibroblasts, and the lowest is about 65% for the human stem cells. So overall, we have a very good mRNA transfection efficiency. Then we dig more deeply, like we want to know that whether it's possible to control the number of mRNA that we deliver into the cell. As I mentioned previously, the major mass transporting mechanism is through diffusion and electrophoresis. So the total flux of the molecule to transport is the diffusion plus the electrophoresis. And we also know that, as I mentioned, for charged molecules, the electrophoresis is going to be the dominant because it's much more faster, and then the equation will be the total flux will be approximately equal to the flux from the electrophoresis. So if we do the calculation or do some math, we know that we got this equation. So the total number of molecules to get into the cell will be equals to the nanostraw opening times the electroosmotic the electromobility of the mRNA or the cargo molecules, and the field intensity, the electric field strength or intensity, and the cargo concentration. and the delivery duration. And if you notice that, I don't know if you notice that the last three parameters are the parameters that we can tune. So basically, we can manipulate the three parameters to control the number of molecules that we can send it into the cell. And but one thing is to notice that electric field is very hard to change, because if we increase it, it will change the local pH, which can be toxic to cells. So eventually we just manipulate the last two. So just to check, to confirm our calculation, we delivered the sci-fi labeled DNA oligos into the cell, into the HeLa cells with series of DNA concentration and the delivery duration. As illustrated in this cell images, the DNA fluorescence intensity that inside a cell is increased with the increase of cargo concentration and delivery duration. We also plot the relationship. You can clearly see that DNA concentration in the cell has linear correlation with the cargo dosage concentration and delivery duration, which exactly match the equation that we proposed just now. So besides the DNA oligo, we also delivered really has not exactly, but has some biological meaning. The mRNA expressing the mCherry expressing mRNA into the cell and analyze the protein expression in the cell. It is easy to see that as the dosage of the mRNA increases, the protein expression in the cell increases. We also find that the linear correlation between the dosage of mRNA and the protein expression level in the cell. So we're talking about delivery. So then the question is, if the Nanostraw allows direct delivery, or allows us to deliver the molecule into the cell, does that mean that we also can extract the molecules from the cell through the Nanostraw? So the question you may ask is, why do we want to do that? The conventional way to interrogate gene expression in cells is that we have to tear the cell apart to collect the intracellular content for analysis. The issue with the current method is that we only could just get a snapshot of the current state of the cell, and because the cell is dead after analysis, we cannot know the future development of the cell. This is more like take a picture of a person. If you really look at the picture or the snapshot of the person, you cannot really predict what the person is doing, right? So in order to accurately monitor the cell and understand the cell behavior over time, what we really want to do is to take a movie of the same cell over time. That means that we cannot kill the cell for analysis, and we need a system to allow us to do the living cell biopsy. From the living cell extraction really represent the gene expression in cells. We compare the living cell extraction with the same cell lysate. In this experiment, we randomly select 48 genes and use qPCR to quantitatively analyze the gene expression level of each gene and plot. And here we plot the 48 genes expression level of the living cell extraction and the cell lysate. A split plot shows that there are 41 genes in the living cell extraction has strong correlation with the gene in the cell lysate, which suggests that what we extract is from the living cells has very good representation of the intracellular gene expression. However, we do find that there are 7 outstanding genes that has a statistical difference from the cell lysis, and this is labeled under the asterisk sign. And we hypothesize this is due to the lower extraction efficiency of the 7 genes, and the inefficient extraction could be caused by several reasons, including low diffusion rate resulting from the mRNA size or the gene sublocation within a cell. But we do not know exactly why causes. It's just hypothesis. So the Nanostraw system not only allows us to do living cell extraction, but it can do it repeatedly. So in this study, we showed that we verified that we can do repeated extraction of the mRNA from the same set of beating cardiac cells over 3 days and analyze the mRNA extraction level of the 18 genes in the extraction. So here's the first movie to record expression level in the same set of beating cardiac cells, which is right here. And for most of the gene, the gene expression level is pretty consistent, and that's actually demonstrates the precision of the extraction analysis process. But notably that there are some interesting gene fluctuation, and as for what causes this fluctuation, we do not know, and nobody knows why there is fluctuation. So there's more study needs to be done. We need to do more analysis. So you summarize the first part of the talk. I introduced a Nanostraw system that allows the formation of direct intracellular access and realize dosage controllable longitudinal delivery and extraction of living cells. And because the unique feature of Nanostraw, the system really provides a innovative methodology to promote longitudinal gene expression and monitor of the cell behavior in a closed loop fashion. And there are many biological research studies that we believe that can be greatly impacted by this system. For example, here I list the 2, which is in the field of induced pluripotent stem cell reprogramming and stem cell differentiation. Yeah, so this is the what I talked is about Nanostraw, and then I would like to introduce the second part, or the second system that I developed during my postdoc study training, which is nanopore electroporation. Yeah, so just like what I mentioned in the introduction, that electroporation is a relatively toxic process or method, and the main challenge of it is that it forms too much pores on the cell membrane. The bottom right plot is the calculation of that predicts the number of pores formation on the cells caused by the typical electroporation, and the dashed line showing right here is the typical electric fields that would normally use to porate the cell. As shown in the plot, we use the typical fields right here that if we use typical field to porate the cell membrane, there are about 7,000 pores formed on the entire cell membrane, and this is definitely can be lethal to a cell. So just give you a better idea, I find another video that really records the cell morphology change during the electric field electroporation. We find that so notice that the cell morphology changed greatly when it experienced a pulse of electric field, and even if the cell recovered after the electroporation, the morphology of the cell is not the same as its original state. So the idea we have is that whether it's possible to limit the electric field, to lock the electric field, localize the electric field at a very small spot or a nano area spot. So the only area that the cell can experience electric field is at the nano size area, and the rest of the the cell membrane will still remain intact. Can we do that? We also verify our idea by doing a console simulation to look at the actual electric field distribution through a nanopores. So if in this model we created two salt reservoir right here, and then use a nanopores to directly connect these two reservoir. And then we applied voltage potential across the nanopores and analyzed the electric field distribution. And this is the plot. Let me see right here. This is the plot of the electric field intensity along the tray line, the trace that moves away from the nanopores. And the red dashed line showing in here is the typical field strength to porate the cell membrane. And there are two things I want to point out right here is that the nanostructure, by using this nanostructure, we only need about 20 volts to generate enough electric field at the local area to porate the cell membrane. In comparison with the normal cell electroporation, which is about which can be from 300 to about 1,000 voltage per voltage. This is greatly reduced. And the other thing is that if you look at this carefully, the electric field intensity drops quickly as it moves away from the electric field, from the nanopores, and that's actually and this suggests that membrane and actually this is really important for protecting the cells, as that means that even with 100 nanometer, even the cell membrane that is 100 nanometer away from the nanopores will not feel strong electric field and will remain intact. Yeah, so we have this theoretical calculation. Then the next question is that, can we make it? How do we make it? What kind of materials we need to use, and what kind of fabrication we need to use. It turns out that the nanostructure is very easy to find. It's just a nanostructure that embedded in the simple water filter. If you look at the surface of the track etched membrane under the SEM, what you see is that the nanopores are evenly distributed on the membrane, and we can just really take the advantage of the water filter nanogeometry to build our nanostraw or nanopore electroporation system. So to make this device, we simply take a piece of water filter and assemble it with PDMS cell holder. And then if we want to do electroporation, we just simply put the cell solution in the cell reservoir and let the cell to precipitate down to the surface and making good contact to the nanopores. And then when we want to do the electroporation, we just simply sandwich this device between the electrodes. Normally I use titanium. You can use other electrode to do the delivery. So to test the efficiency, we have pretty good result. Right here we delivered mRNA and also DNA into the cell. Right, this one is mCherry expressing mRNA that transfected the Jurkat cells. So notably that we at under a voltage of 30 volt, we have pretty good transfection efficiency, which is about 80%. And the other important things to note is that even if that we have higher voltage, the cell viability always remains in 95%, which just as what we expected. So the nanopore electroporation not only allows DNA nucleotide delivery, but also various kind of protein delivery. This one is we demonstrate it's able to deliver proteins, various type of protein, and right here is the RBD proteins delivered into the primary T cells with high efficiency. And we also demonstrated that the T cells, primary T cells, after the electroporation has very preserved its activity. And this is the T cells that after the delivered protein delivered T cells after delivery, we directly put them into the lipid bilayer and allow them to integrate with the lipid bilayer and monitor their activity. As you can see that the T cell is actively searching for antigens. It moves very fast, and that indicates a good sign that the T cell still under very good condition. And the capability of the delivery proteins into cells is actually very useful features that has many biological potential applications. One exciting applications that we find is to use the system to promote multicolor single molecule tracking. So if you are not familiar with intracellular or single molecule tracking, just give you some background. Single molecule tracking is a powerful tool in microscopy, and it allows monitor the fluorescence molecules labeled particles such as proteins in a cell, and provides important insights into protein dynamics, regulation, and functions. And the standard protocol to perform single molecule tracking is not hard. And so we first need to genetically tag introduce a tagged protein, target protein, and then we can incubate, allows the protein to express, and then we incubate the transfected cells with cell membrane permeable dye. So allows this permeable dye to tag the targeted protein, and then after the incubation, we can wash the cell and send it to the microscope for imaging. And this standard protocol is robust for tracking specific protein. However, if we want to analyze two or more type of protein interaction, the protocol is not practical. And I will tell you why it's not practical. So this is the current limitation for protein labeling. There are three major challenges. One is that if we label too much different type of protein with more than two cell membrane permeable dye, we have problem. The one is that the cell membrane permeable dye normally will interact with cell membrane. That means that normally if we label multiple dyes in a cell, we will have a lot of background noise on a cell membrane. And the other is that there are sometimes low labeling efficiency and specificity can be happened. Then that means that if we do labeling multiple proteins, we may introduce mislabeling. And then third one is that there are really not a lot of commercially available high quality cell membrane permeable dye that we can use. So because of these limitations, that makes us want to think that whether it's possible to do that, to use nanopore electroporation to promote multicolor single molecule tracking. The idea we have is, why don't we just label the proteins, purify the proteins that we want to image or we want to analysis outside the cell, and then do the labeling outside the cell, purify the protein and do the labeling outside the cell, and then send the labeled protein into the cell through nanopore electroporation. And this is the fundamental idea, and we did and then to really analyze our idea, we have a model. We want to or the pathway we want to analysis, which is the epidermal growth factor receptor pathway. It is a very normal and well-known pathway. People have studied many years, and what we're looking at, what we're interested in this pathway is these three molecules: Grb2, source, and RBD. And we basically purify these three proteins and use nanopore electroporation to deliver these three proteins and see whether they have biological activity interactions, and whether it's actually we can monitor them simultaneous in a single living cells. I will, because of the time, I think I I talk too much. I will not talk about this too much, but just want to show you that yes, we are able to deliver the Grb2 into the cell, and the Grb2 is actually response to the EGF when we present the EGF to the cell. The Grb2 actively responds. Basically, they are activities that Grb2 will attach on the EGFR rapidly after the EGF stimulation. And also we are able to monitor two color like the Grb2 and source, and we are able to analyze their interaction with time resolution. Like right here, this one we show that Grb2 will first recruit on the surface on the receptor, and then the source will attach to the Grb2. So there is the time difference. There's a delay for the source to recruit on the surface. Yes, and we are able to do 3. If we have a good quality dye, we can do 3, 4, 5, you name it. So to summarize my talk, I particularly introduced one of my major aspect of my research, which is in vitro intracellular delivery. Of course, I have the other 3. I'll quickly go over. For oral delivery, we make the micro device that allows us to pierce the intestine, the surface of intestine, and allows us to release insulin or protein peptides. Of course, this is just a demo. We in my lab in the future we will optimize the surface and do and really trying to commercialize this nano micro device. And also for MEMS, I'm an expert in nanofabrication, so we are able to use MEMS to do a lot of different nanostructures. Really, that what I want to do is trying to build a high throughput micro injection. So instead of using this kind of micro injection that has low throughput, and you have to need a technician to pierce the zygote, what we want to do is using this chip to do high throughput micro injection. So then eventually we don't need a high sophisticated equipment and technician to do that. Then the other important aspect I want to mention is the using nanoparticles or other magnetic nanoparticles to do genetic engineering of plants. Right here we show that we are able to use nanocluster to carry the sense RNA and directly inject it to the plant tissue to downregulate gene expression in plant. We also have a collaboration with one of the plant experts in China that we have a project using magnetic to engineer pollen, plant pollen, to use this pollen to carry genetic informations like plasmid, and use this one to use the pollen, the DNA carrier to transfect the actual cotton in order to manipulate, really do the gene engineering of the plant. So other than that, that's the end of my talk. I just graduated, and so I would like to thank my advisor, Nick Moulash, and also my collaborators at Stanford and UCSF, and also my postdoc advisor, Peradeniya and Stephen Chu, and also my collaborators. And please let me know if you have questions. And this is my my information. If you want to join me, please contact me. Thank you.
Yeah, clap, clap. Yeah, thank you, Dr. Yuhong. So as much as there's a lot to explore if we look up, and there's a huge universe to explore, and there's same manner, there's a universe to explore if we look into ourselves, into our cells itself in the nanoscale. And thank you for giving us that insightful talk within this short amount of time. And you covered the two concepts of Nanostraw and the nanopore work you did in a comprehensive manner. And I guess we have time to take up some questions from the audience. And we have a couple of questions in the chat. And so I would like to invite the audience, if you have any questions related to her research, as well as I think she'll be happy to answer if you have any questions, if you have any general questions related to a research domain or the work she's doing. And I see that there are a couple of questions that are specific to what you presented today. Sorry about that. So, so I guess we can move to questions. Is that okay, Dr. Yuhong?
Yeah. We can take up the questions. Yes.
Yes. So
yeah, I read there are questions in the chat window.
Yes. Yeah, that's correct.
On the cell. So the first question I see is, what will be the effect of electrophoresis material on the cell?
Yep.
Yeah, what would be the effect of electrophoresis material ion? Okay, on the cells. Yes, the electrophoresis is definitely gonna have some effect. Specifically, the cell, the concentration of ions has the cell particularly holds a certain concentration of ions like calcium, potassium. So if we use electrophoresis, definitely it will cause cell the concentration loss or imbalance in the ion concentration loss or imbalance on the inside the cell. So this is the things that we cannot avoid currently, because we need no matter how, we need to have pores open a hole on the cell in order to introduce the molecule into the cell. But yes, it has some effect, but currently we do not notice a significant, like notable statistical difference. We didn't find any currently. And the next question is, can we only send negative charged molecules across the membrane? No, you can do negatively charged, positively charged. All charged molecules are fine as long as you reverse your electric field, right? But there are chance for now no charged or charge neutral molecule. You can also deliver charge neutral molecules into cells by diffusion. As I mentioned, diffusion is another mechanism for mass transporting, but it's certainly not as efficient as the charged molecule. And okay, are the cargo molecule polarized for yeah, we do not polarize any molecule. If the molecule is charged, it is charged. We cannot do much unless we modify the cell by say by labeling the cell with highly charged something, like highly charged dye or highly charged nanoparticles. You have to engineer it if you really want to make it polarized. But in the particular experiment that we did, we don't. It's just naturally what the molecule is. We just simply use it. And what is the method that you use to measure n value with concentration and time? And specifically, what does n value mean? The number of what, cell? Or I'm not sure about this.
Yeah, I think this question is referred to the slide number 13 in your presentation. Maybe the equation, that's the question, I suppose, which has been asked. N equals A mu E C0T. Yeah.
30.
13. Yeah. Oh, sorry, 16.
13.
16. Yeah.
17, right? Or the dosage control delivery. Is that correct?
Yes, yes.
And then n value, or okay, I see. So let me see, measured n value. So right here, where the n value in the equation, the n value is the number of molecule that we deliver into the cell, one cell, and and the measurements that we did in this particular experiment is that we measure the intensity, because the intensity, the fluorescence intensity it's directly correlate with the number of molecules. Is that correct? Is that clear? That so right here we have we cannot count the number of molecules in cells, right? It's too small. So we use fluorescence as a representation of n, the number of molecules.
Yeah. Actually, Dr. Cao, I think we have another question from Dr. Suranga. So yeah, he's from the Faculty of Agriculture. So over to you.
Hello, Dr. Cao. It's a nice presentation. I must appreciate your talk. Just one question, because I'm a reproductive biologist, so I'm interested more in something you told about oocytes. So after this, your electrophoresis, have you ever looked at their the developmental ability, like fertilization and subsequent things in your experiments? I've seen you have devices, small micro injector. So have you ever tried, I mean, after injecting, how was the developmental potential of the oocytes?
You mean a zygote? Is that correct? The fertilize,
the
egg, right? Yeah. So we're making the device. We're not really having. We have a now it's more like in a immature state. We're not really have the device is not ready right now. It's still under development. We didn't do much more biological experiment yet. We haven't.
Oh, okay. Thank you very much.
So Yuhong,
go ahead.
Yeah,
go ahead.
I have a kind of a general question on behalf of the audience we have today, because we are having students also participating in our session today. So can you give some insights about what sort of higher studies opportunities are there related to this research field?
Sorry, I didn't catch. What are the
what sort of opportunities are out there in terms of pursuing higher studies in this field?
Yeah.
Oh, opportunity in terms of pursuing the study.
Yeah, if someone wants to do a PhD in this area, let's say.
Right. Yeah, so it is a interdisciplinary, very cross-disciplinary field. It's I mean, the delivery, the intracellular delivery is too broad, and you basically can apply it to all kinds of bio-related research. So if you want to use it, develop a method and use it for your research, is a very interesting pathway for career in terms of career development, or there are other for industry. So everything that you built during your PhD it can be a product. I mean, this may not be, or it can be. You have to if you want to pursue industry, if you want to turning into a product, or making money, there are chances that you can do it. I know there are a few professors who have developed systems, the delivery system, and commercialize the system. So the opportunities are pretty broad. It depends on your interest. And also that in this field, like I said, if you want to do nano or microfabrication using lithography, we have a lot of opportunities or trainings for you. And the microfabrication is a very good technology that a lot of people wants to acquire. And because when you trained, you can go to the semiconductor industry. That's another very big industry. So it depends on the students' career projection.
Yeah, so thank you, Dr. Cao. So I have another question. So what kind of PhD opportunities are available under you? I mean, do you have any particular PhD opportunities with you right now? I think our students might be very interested to know about such opportunities as well.
Yes, I have. I'm open. I welcome the students, the talented students who are interested in delivery, nanofabrication, plant, and cell engineering, gene engineering, like using CRISPR. That's all the aspects. I mean, we can discuss, like, there are a bunch of interesting projects that I can think of that we can do. Of course, there are other things, like I'm also open to hiring postdocs. I have postdoc program. So if there are people who are almost like ready to graduate, I'm also like willing to talk with the senior graduate students.
Okay, so thank you, Professor Cao. I think if Professor Wei is around, maybe he would be able to tell us a little bit about the procedure of recruiting students, because yesterday also we had a very interesting session on the ANSO scholarship scheme. So Professor Wei, if you could share with us a little bit about this PhD recruitment procedure with us, that would be great.
Yeah. So many thanks. And yesterday afternoon, so can you hear me? Yeah.
Yes, sure.
Yeah.
Okay. Yeah. So it's really wonderful, the present lecture. So thanks Professor Yuhong Cao. So, and also thanks the UOP International Affairs Office to organize such the webinar. So, and I think so for the PhD starting to in China and in class. So this is a very good opportunity, because Professor Cao just come back from USA to China, and there's some opportunity for the young talents for master and the PhD. So I think if some students are interested in Professor Yuhong Cao's research area, please send email to her, and I think it will be much better. So, and maybe next week it's the same for us. So the next week we will give you a short list name. Then we'll confirm, and then it's okay. Then they will, and then we'll for a short name list, and then we'll decide who we will be accepted to apply for this ANSO scholarship. That is our procedures, because you know the time is a little bit limited. I hope your young talent can catch this opportunity, because yesterday, so we focused on the water and the environment. This area I totally don't know. So that is why I would like to invite Professor Yuhong Cao to make a presentation to show her research and field, and that will be good to make a communication with students and colleagues, and then I hope also can set a bridge to cooperation in the future. So that is so. Thank you. Yeah.
Thank you.
Yeah. So thank you, Professor Wei, for your thoughts, and thank you, Dr. Yuhong Cao. So I will get back to Dr. Damayanthi again. So it's time for you to make some closing remarks, and finally, I will give some thanking note as well.
Yeah, so as I mentioned earlier, we had a very productive session, and thank you so much for giving that comprehensive talk during that short amount of time we had today. And there were a couple of questions that they were not answered, but I think the International Affairs Office and we can coordinate with the audience if they have any further questions and they want answers for them. Yeah, thank you Yuhong again. And look forward to working with you possibly.
Yes. So
yeah, of course.
Thank you so much, Dr. Damayanthi, for being here with us in moderating this session today. And so let me make a quick thanking note to everyone who participated. So Professor Shamini Jindaswar, Director of the International Relations Office, then Dr. Kamalnath Samarakon and Dr. Damayanthi Herath, representing the Faculty of Engineering, where we hosted this program. And a very big thank you to Professor Yuhong Wei and Dr. Yuhong Cao for all your efforts in promoting the scholarship opportunities for the students at Peradeniya. So we look forward to bringing students this year as well. So last year has been very successful. So we hope that students very much interested will be joining with you as well, Dr. Cao. So we will always be in touch with you and with the students also. So any prospective students will be, so you can send us your details to International Relations Office. We'll be very happy to connect you with Dr. Cao. And regarding the answer scholarship, if you have any questions, you are most welcome to contact us via email, or you could physically visit us. So with that, I would like to bring an end to this program. So thank you, ladies and gentlemen, for reserving your valuable time. So we look forward to meet you again in another program like this. So take care and stay safe. Goodbye. Thank you.